proteins. So here proteins are polymers of amino acids. Polymers of amino acids. So here the monomer of protein molecule are amino acids. So the amino acids undergo condensation polymerization to form a polymer through the formation of amide linkages. So where the amide linkages are known as peptide linkage. So that's why the resulting polymers are known as polypeptides. So as all that the resulting polymer molecules consisting that the peptide linkage, they are known as polypeptides, and the polypeptides those which are having molecular mass more than 10,000 are known as protein. If the molecular mass of polypeptide below 10,000, they are generally considered as peptides. Okay, of course, the peptides proteins are nothing but polymers of amino acids, but depending upon the variation in their molecular masses, some of the polypeptides are considered as simple peptides and remaining polypeptides are considered as proteins. Now let us discuss the structure of protein molecule. So protein molecule consisting a long chain linear structure composed of various amino acids. So here we have to study the structure of proteins on the four headlines. One is primary structure, second one is the secondary structure, next tertiary structure, and the last one is the quaternary structure. So here these are the four headlines under which we are going to know about the structure of protein. So, primary structure, secondary structure, the third one, tertiary structure, fourth one, quaternary. Each and everyone can give a particular information regarding the structure of the protein. Now let us consider initially the primary structure of protein. So here that the primary structure give an idea about the nature, number and sequence of amino acids present in the peptide chain. So here you will get the most important information in the primary structure. So majorly it gives an idea about what type of amino acids are there along the peptide chain and what is the sequence of that amino acids. Okay, if suppose if that protein molecule or peptide chain composed of three different amino acids like ABC. So what is the sequence of that amino acids? What is the nature of A, what is the nature of B, and what is the nature of third amino acids? Okay, so here we are going to get the beautiful information about the sequence of amino acids in the peptide chain. So in a particular when you draw the molecular structure of a general protein molecule or a polypeptide, so you may know that both ends of that given polypeptide chain is not the same. On one hand, we are having a free carboxylic group. On the left hand side, we may have a free amino group. So let us note down the polypeptide chain. Like this. So already I have given this general structural formula for polypeptides. So see here, both ends of the peptide chain are not the same. On right hand side, we are having the free carboxylic group. On the left hand side, we are having the free amino group. So here, this left hand side amino acid is known as N-terminal amino acid, and the right hand side amino acid containing the free carboxylic group is known as C-terminal amino acid. So, in the primary structure of proteins, we have to determine the N-terminal amino acid, like that C-terminal amino acid, and what type of amino acids are like in between that of both amino acids. Okay. So further, initially determine what is the N-terminal amino acid, followed by determination of C-terminal amino acid. So for that, we must follow a particular method. So here the two most important methods are there. One is Adam's method to determine N-terminal amino acid, and Slack-Poof method to determine C-terminal amino acid. Now let us discuss the first one. Adam's method. So here the Adam's method is useful to determine N-terminal amino acid. N-terminal amino acid. So now let us discuss how the N-terminal amino acid will be determined in the polypeptide chain. So here that polypeptide should be treated with phenyl isocyanate in the presence of alkali solution. So let us see the various steps that are involved in the Adam's method. So C6H5 So this is the phenyl isocyanate. Now treat polypeptide chain with this reagent NH2 CH or CO then HCH CO continuation is there. Okay. So depending upon the number of amino acid residues present in the peptide chain. Okay. So here we are majorly highlighting that amino acid residue in the polypeptide chain having free amino group. So here the amino acid residue containing this free amino group involved in a reaction with the reagent and it gives phenyl carbamide peptide. So now let us see. Alkali substance acts as a reagent. In the presence of that reagent, these both reactors react together and give a resulting product RCH H. So the resulting product is known as phenyl thiocarbamyl peptide. Okay. So this compound undergoes dehydration in the presence of mineral acid and give a 5-membered heterocyclic compound which is known as thiazolinone. Is known as a thiazolinone. So doing that, the rest of the peptide chain will be removed by means of hydrolysis in the presence of the mineral acid. So here we will get R, C, H, then double bond C. Plus remaining H2 then R, right? So this is what we got after the second step, and that means that the resulting phenyl thiocarbamyl peptide conjugated with the mineral acid it gives thiazolinone. Okay. Now that the thiazolinone on hydrolysis it gives the amino acid residue R, CH, then H, COOH, CS, NH, CCH. So this is the resulting the product. That means the N-terminal amino acid has been separated as phenyl thiocarbamyl amino acid. So there what we get? Phenyl thiocarbamyl peptide. So after treating the mineral acid, so it forms phenyl thiocarbamyl amino acid. So which undergo dehydration immediately in the presence of mineral acid and it gives phenyl thioiodine ion or CH. Then H, C, S. Phenyl PTH. So this is a resulting cyclic compound. So that PTH will be separated from the reaction mixture and identified by means of chromatographic technique. It may be either column chromatography, layer chromatography, so whatever it may be. Okay, so you use either column chromatography, layer or paper chromatography technique. Separate that phenyl thioiodide ion from the reaction mixture and identify what is the N-terminal amino acid. Of course, when we treat that phenyl thioiodide ion with the weak base, then automatically it gives a corresponding to amino acid. NH2 CH or CO. Okay, so this is the resulting amino acid that will be separated and identified by either of the three chromatographic techniques like column chromatography, thin layer chromatography or paper chromatography. So this is the over where we determine what is the N-terminal amino acid in the given polypeptide chain. Of course, it is also possible to determine the C-terminal amino acid by following another method. So now let us see how that C-terminal amino acid is determined in the given peptide chain. For that, we are going to use SCLAC and Simpson method. SCLAC and Simpson method. So one of the most important technique to determine C-terminal amino acid in a given peptide chain. Of course, so before going to treat the given peptide chain, it is our duty to protect the free amino group of N-terminal amino acid by means of benzonylation. So now let us take that for given polypeptide chain. So this is the given peptide chain. Let us consider it may be like this. So again here we are having the amino group, free amino group and the free carboxyl group on both opposite ends. So before going to treat this peptide chain with the respective reagent, we have to protect that amino group. So to protect that amino group, generally we use benzoyl chloride. So treat that compound with benzoyl chloride. C6H5CO. So what happens? That the free amino group involved in the condensation with the reagent on the form N protected polypeptide chain. Okay. So of course you have to use a strong oxidizing agent for the condensation. In the presence of that strong basic substance, so we can expect the loss of HCl, So that strong base can extract the acid moiety from the two reactants. So here it is ready to use chlorine as chloride ion, and this amino group is ready to convert this hydrogen as H+. So totally there is a loss of the acid moiety in the presence of the strong base, and it gives the N-protected peptide chain C6H5 NH CH or CO. So this is the unprotected peptide chain. Sorry. So here we have to write C. C6H5CO, NHCHR, CO, NH, CHR, right? So remaining is. So peptide linkage CHR double bond CO. Okay. So this compound now treated with ammonium thiocyanate. Ammonium thiocyanate in the presence of acetic anhydride. Okay, so here we have to maintain certain temperature. So at that temperature, what will happen? That ammonium thiocyanate will be converted into thiourea, and then it involves in a condensation with that terminal amino acid having the free carboxylic group with the elimination of 1 mole of ammonia and 1 mole of water molecule. So here let us see. So this will be converted into H2N CS NH. So NH4 CNS. So at particular temperature, so that means when the temperature reaches to the suitable temperature of the ammonium thiocyanate to convert into thiourea. Okay. So in the presence of that acetic anhydride, these two molecules undergo condensation with the loss of water molecule as well as ammonia. So here this NH group and carboxyl group involved in a condensation with the resulting thiourea and form the resulting condensation product where we will get the heterocyclic compound at the right hand side of the peptide cell CH or C4 NH CH or this is the resulting product where we are having the 5-membered heterocyclic compound at the right hand side of the peptide chain. Now treat this compound with the alkaline solution or sodium hydroxide. So in the presence of that aqueous sodium hydroxide, that molecule undergo hydrolysis and it forms thioiodate ion. C6H5CO NH CHR CO NH CHR CO plus NH CHR right C yes CO. This is the thiourea. Okay. So now when we treat this thioiodate ion, when we treat the thioiodate ion with barium hydroxide, then we will get then CHR double prime CH. So this is the C-terminal amino acid present in the given peptide chain. So that will be separated and identified by chromatography technique. Of course, you can apply same C-terminal amino acid determination method on the remaining peptide chain. Of course, in the previous case also, the N-terminal amino acid determination method can be applied on the resulting peptide residue. So whatever that peptide residue remains in that reaction mixture should be treated with their respective reagents in the same way, and you can determine so what may be the C-terminal amino acid on the resulting peptide residue. So you can apply the same a number of times until you find out the amino acid residues that are present in the given peptide chain. So this is the what where you can determine both N-terminal and the C-terminal amino acids by Edmonds method and the Schlatt method respectively. Okay. In the given peptide chain.